This week, we look at the world of plants, their life, medicinal potential, and genetic variety. According to biologists, plants were the first link in our genealogical tree. It was the plants which created the oxygen in our atmosphere. They also converted solar energy into a veritable larder, which led to the emergence of animals on Earth, and later, our own. Together, let's discover some of the secrets of these silent relatives of ours. We tend to think of plants as inferior to the animals that we are. They have no nervous system, cannot move about, or make sounds. And yet, like us, they have to sustain themselves, grow and reproduce. To perform these vital functions, they rely on incredibly complex mechanisms that even animals could envy. A plant's nutrition begins at its roots. These underground organs branch out into fine radicals. The radicals are covered with a coat of minuscule root hairs. The root hairs trap the water and minerals in the soil. This nutritious mixture, called crude sap, then links up with a network of canals that runs through the plant. The sap must be transported all the way to the leaves. This is accomplished by a simple but effective system, transpiration. Transpiration occurs thanks to small pores, or stomata, on the surface of the leaves. At night, the stomata are closed, but in daytime they open to let the water contained in the leaf evaporate. This evaporation draws the crude sap up towards the top of the plant. However, if the air is too hot and dry, transpiration can be excessive and cause the plant to wilt. The plant completes its nutrition through photosynthesis. This process occurs in the cells of the leaves. The cells contain tiny chloroplasts, which in turn contain molecules of chlorophyll in neatly arranged rows. These molecules act like antennas, trapping the light waves of the sun. Simultaneously, the carbon dioxide in the air enters the leaf through the gaping stomata. With the solar energy collected by the chlorophyll, the carbon dioxide combines with the water to form glucose and oxygen. The oxygen is quickly expelled through the stomata. For plants, it is basically waste. As for the glucose, it is transported to the plant's various parts, where it will be used to make a myriad of compounds, oils, fibers, starch, proteins, etc. Through photosynthesis, plants produce the nourishment they need to grow, and in turn, their slow but steady growth affords them greater access to light. The impressive sizes plants sometimes attain are due to a particularity of their cells. Plant cells are always encased in a rigid wall of cellulose. This frame is sometimes enriched by other components, such as lignin. Lignin is the chief constituent of wood and is found in varying degrees in most plant species. Like all organisms, plants need to reproduce. They can do this in one of two ways. Vegetative propagation enables the parent plant to clone itself, that is, produce identical replicas of itself. For example, certain species propagate creeping stems, or stolons, that root and produce new plants. However, sexual reproduction gives plants their best chances of survival. It assures a continuous renewal of their genetic characteristics, allowing them to adapt to changes in their environment. Plant sexual reproduction occurs in a great many ways, but it's in flowering plants that it reaches its zenith. A flower is a group of leaves modified for reproduction. These modified leaves are generally arranged in a series of rings. On the outside, the sepals serve to protect the flower bud. Then come the petals, whose role is generally to attract insect pollinators. As they open, the petals uncover the stamens and the pistil, respectively the flower's male and female organs. The stamens release the pollen that is transported from one flower to the next by the wind or by insect pollinators. Each pollen grain contains 2 male nuclei. When a pollen grain settles on the end of a mature pistil, it produces a very fine tube that penetrates to the ovule located in the ovary at the base of the pistil. The tube carries the 2 male nuclei to 3 of the 8 female nuclei contained in the ovule. Double fertilization then occurs. One of the ovule's fertilized nuclei begins to divide, forming the embryo of the future seed. Then 2 of the ovule's other nuclei fuse and are in turn fertilized. They will form the albumen, a nutritive reserve for the embryo. Meanwhile, the wall of the ovule thickens to form the seed envelope. As for the ovary, it turns into fruit. Seed is probably one of the plant kingdom's greatest achievements. Not only does it contain all the elements of the future plant, but the nutrition they need to develop as well. It is therefore able to regenerate them at all times. In addition, its ability to disseminate from a distance amply compensates for the immobility of the parent plant. Botanists believe that in large part, it is this modest organ that accounts for the amazing success of seed plants. The vegetable kingdom is not only a larder, it's a pharmacy as well. Our ancestors found it to be a treasure trove of natural remedies. Unfortunately, treating disease with plants was associated with witchery and thus abandoned. Today, scientists are rediscovering the tremendous healing power of plants. One hardly feels close to nature when browsing through the aseptic shelves of a modern pharmacy. But appearances can be deceiving, for no less than 60% of today's drugs derive, at least originally, from the plant kingdom. Indeed, plants constitute an immense reservoir of substances with therapeutic properties. Every year, medicine uses many such substances to create new drugs to fight serious diseases such as cancer. But also, in recent years, the often forgotten medicinal properties of entire plants are being rediscovered. Medicine draws a great deal of its knowledge from traditional pharmacopoeia. For example, a long time ago, our ancestors noticed that the bark of willow trees had analgesic, anti-inflammatory properties and helped to reduce fever. It was later observed that what accounted for these properties was the presence of acetyl salicylic acid, a compound that was given the name of aspirin. Today, synthesized in the laboratory, aspirin has become the world's top-selling drug, but we may never have thought to use it had our forebears not made the observations they did. The medicinal components of plants belong to various chemical families. One of the most important is the alkaloids, characterized by nitrogen atoms in their chemical structure. Some alkaloids, like atropine from the black nightshade, and nicotine from tobacco, are toxic. Conversely, the alkaloid morphine from the opium poppy has become indispensable in treating acute pain. Quinine, derived from the bark of tropical America's cinchona tree, is highly effective in treating malaria. Vincristine, obtained from Madagascar's periwinkle shrub, has saved thousands of children suffering from leukemia. This alkaloid combats cellular division and thus prevents the multiplication of white blood cells that is associated with this type of cancer. Unfortunately, it takes 15 tons of periwinkle leaves to produce a mere 30 grams of vincristine. To get around this problem, some biologists are attempting to produce vincristine through in vitro cell culture. This is no mean task, since only the leaf cells of the periwinkle produce vincristine, and then only at a specific stage in their development. Nonetheless, the researchers are beginning to understand the cellular mechanisms involved in the synthesis of vincristine. They therefore hope to create, through genetic engineering, cell lines able to produce large quantities of the drug on an ongoing basis. Although some active plant substances are already abundantly used in medicine, we still have a long way to go before they have all been inventoried. New drugs are constantly being found in rainforest plants, as they are in our own garden plants. For example, it was recently discovered that the yew, an evergreen often used as an ornamental plant, contains a component highly active in fighting certain forms of cancer. Also, researchers are working to make an inventory of the active agents of plants and find out how they affect our health. The components are isolated in the laboratory by thin layer chromatography. This technique makes it possible to separate the various components of a plant and discover their relative proportions. To do this, the plant extract is placed on the base of a glass plate, evenly coated with alumina or silica gel. The glass plate is then set vertically in a solvent bath. The solvent moves up toward the top of the plate, moving the chemical components of the plant to different distances from the base line, depending on their individual physical and chemical traits. These components are made visible by spraying a reagent on the plate. Some substances will show up when the plate is viewed under ultraviolet light. The exact location and intensity of the spots, precisely measured, enables the researchers to identify the plant's different components and calculate their proportions. Medicinal plants used to be gathered in the wild, but these natural sources are now threatened. So concurrently with their lab work, researchers are trying to cultivate certain species in order to determine optimum growth conditions. Laboratory testing also helps to identify the parts of the plants which are highest in active agents, as well as the most suitable times to pick them. We are learning more and more about medicinal plants and their uses, but the researchers also see ancestral knowledge as a valuable source of information. Unfortunately, traditional ways are gradually being lost, and in addition, many plant species are vanishing from the earth. These traditional remedies must be recorded for posterity before they are lost forever. By subjecting plants to their own needs, human beings ventured onto slippery ground. Many cultivated plants which have reproduced among themselves for generations are as fragile as inbred children. Fortunately, scientists are now endeavoring to preserve plants' genetic variety. Many countries are witnessing the emergence of new kinds of banks. These new bankers are botanists and geneticists. Their customers are farmers or horticulturists. Their vaults contain an unconventional treasure, thousands upon thousands of samples of crop species. Seed, the genetic inheritance of crops, is an extremely precious commodity. It enables us to develop new crops that are more productive, more nutritious, and more resistant to disease and drought. In the long term, our seed banks are vital if we want to keep our fields from turning into deserts. The genome is the complete set of genes of a species. Located on chromosomes in the heart of cells, the genes constitute a sort of dictionary in which the characteristics of every species are encoded. The genes control, for example, the development, growth, and reproduction of plants. They also endow them with specific advantages, such as resistance to disease, tolerance to frost, and high yield. During the course of evolution, every plant species built up its own collection of genes. Moreover, the different populations of a same species often develop distinct traits encoded in their genes. It was by exploiting this genetic variety over millennia that man created today's crop plants. By subjecting wild plants to painstaking selection and cross-breeding operations, we have obtained crop varieties that better meet our needs and the requirements of agriculture. For example, the wild cabbage gave us such diverse vegetables as head cabbage, Brussels sprouts, cauliflower, kohlrabi, and broccoli. Similarly, wheat is the result of countless crossings of wild grains from the Middle East. This evolution produced more resistant wheat heads with bigger, more compact grains that are easier to harvest than their wild ancestors. However, in the past few decades, the genomes of plant crops have begun to erode, a phenomenon the scientists have dubbed the inbreeding effect. One of the reasons for the inbreeding effect lies in the small number of plants man has chosen to cultivate. Out of some 250,000 known plants, we rely only on about 30 to fulfill our dietary needs. Also, these few varieties are often monocultured. This means that millions of acres of land can be covered with a single genetically uniform crop. This practice does maximize the yields, but it makes the crops extremely vulnerable to disease and insect pests. Intensive agriculture may also have entailed the extinction of numerous wild varieties that, while not very productive, may have possessed valuable genetic qualities. Many of the wild species that gave us our current crops probably no longer exist. Gene banks will help preserve what remains of that inheritance. They contain thousands of seed samples from the four corners of the earth. Such preservation is possible due to seeds' incredible longevity. When stored in dry and cool conditions, the metabolic activity of the embryo inside the grain ceases completely. The embryo is, in a sense, in a state of suspended animation. The samples are first dried in cold rooms at very low relative humidity. They are then stored in hermetically sealed envelopes. These envelopes can be frozen at -20 degrees Celsius in freezers. Better still, they can be preserved in liquid nitrogen at -196 degrees Celsius. Seed is expected to live hundreds of years under these conditions. However, to be on the safe side, seeds are periodically subjected to germination tests. In addition, the samples are regularly grown out in greenhouses or in fields to allow them to produce new seed. Gene banks are extremely useful Besides helping to preserve endangered plant species, they are made available to plant breeders. Thanks to a computerized catalog, all the data on each sample is readily accessible. In the event of a pest epidemic, a variety that resists the pest can be located quickly and crossbred with the threatened variety to improve its resistance. Today, the data contained in gene banks is used by a veritable army of plant breeders. However, given the pace at which the natural habitats are deteriorating, the race against the clock is far from won. A world without plants would be a very sad place indeed. Actually, deprived of food and oxygen, we wouldn't even be here to discuss it.